Well, hello everyone, and welcome back to Science in the Age of COVID-19. Today we are absolutely thrilled to welcome Paul Bieniasz back to the show. Paul is an HHMI investigator and professor at Rockefeller. Paul's lab has been one of the absolute powerhouses during this pandemic, cranking out results on every aspect to do with the virus and its evolution and prevention. We had Paul on the show about 8 months ago, and he said that we were likely to see a wave of variants coming out in response to, following evolution in humans and perhaps even in response to our various interventions. Unfortunately, Paul was right, and he's back on the show today to tell us about where we stand with the evolution of the virus and how our defenses are holding up. Okay, Paul.
All right. Well, thank you very much, Lauren, for the introduction and the invitation to come back. Let me just share my slides before I get going. All right. So as Lauren said, things have changed since the last time I spoke to this audience. And so what I'm going to talk about today is a little bit about how that change has occurred. SARS-CoV-2 variants and the potential role of antibodies in driving that variation, and some comments about how antibodies might eventually help protect us from these variant viruses that are arising. So let's begin with just a primer, get us all up to speed on what we're talking about. We have been working essentially since the beginning of the pandemic on neutralizing antibodies. Those are antibodies that recognize the SARS-CoV-2 spike protein, which is depicted here, a protein on the exterior of the virus particle. There are a number of important features of the spike protein. It obviously drives fusion of the viral membrane with that of the target cell via interactions between this so-called receptor binding domain, or RBD, and a cell surface receptor called ACE2. And in response to infection and or vaccination essentially involves exposing our bodies to this particular protein, the spike protein that's encoded in all of the vaccines that are currently being deployed. We elicit antibodies to that spike protein, some of which bind to the RBD, and some of those of which inhibit interaction with ACE2. There are other bits on the spike protein that are recognized by antibodies, in particular the NTD, again, when that's recognized by antibody, that can neutralize infection. And there are various other less well-defined at this point areas of the spike protein that can be the target for antibodies. And so the spike protein is constrained in its evolution in the sense that it has to interact with ACE2 and drive viral infection. But at the same time, as we will talk in very great detail about, it's trying to escape the attention of neutralizing antibodies that would otherwise interfere with that process. So let's talk about how SARS-CoV-2 spike evolution might occur. It occurs to me that I actually used a very similar slide to this the last time I spoke to you with the title, Will resistance to SARS-CoV-2 neutralizing antibodies occur? And of course, we now know that that's true. But let's talk about an imaginary transmission chain where we start at this end and end at this end, and imagine the evolutionary forces that are acting on the virus. Of course, the virus is going to evolve, at least in principle, it could evolve to be fitter, to essentially grow better in its host and transmit from one host to another more efficiently. And that at least seems to be part of what's driving SARS-CoV-2 evolution. In addition, as people become immune or are vaccinated, we make antibodies that in principle can interfere with that process. And so a second evolutionary force is to acquire resistance to antibodies. And if we imagine a situation, say in this individual, where a mutation arises that affects one of those properties, the probability of that event happening is affected by a number of different criteria. Some are intrinsic to the virus, what the level of fidelity associated with replication is, the population size and how many such chances the virus gets at making mutations. And also things like transmission dynamics, particularly in the context of antibodies. For a virus to acquire resistance to a particular antibody, it really needs to experience evolutionary pressure from that antibody. And this is a sort of a hypothetical diagram of viral and antibody dynamics. And so we imagine that the virus peaks. Well, we don't imagine, we know that the amount of virus in an individual peaks and then declines, and with it, the probability that that virus is going to be transmitted. Simultaneously, infected individuals are beginning to mount neutralizing antibody responses, but most of the measurements that we've conducted suggest that a large part of transmission really occurs prior to the viral population having extensive experience within an individual of neutralizing antibodies. And so in the early part of the pandemic, there really wasn't much evidence that the virus was evolving in response to neutralizing antibodies. While there was clearly variation, it didn't appear to be directed in any way, focused on parts of the viral spike protein that are targeted by neutralizing antibodies. As I will discuss later, we now think that it's really a little bit different. So the ability of the virus to evolve in response to neutralizing antibodies obviously depends on a number of extraneous factors. So to acquire antibody resistance, for example, in response to evolutionary pressure in this hypothetical individual, the virus not only has to encounter those antibodies, but also has to be transmitted. If it encounters antibodies and that terminates the transmission chain, that's obviously good for us, but it isn't good for the virus in terms of learning how to evade those antibodies. So what features might affect the decision point there? So we know, for example, that in individuals with waning immunity, reinfection occurs at some frequency. We're increasingly beginning to understand that some protection from vaccines, while quite good at protecting us from disease, is incomplete in terms of stopping infection, particularly for some of the less effective vaccines. There are individuals who, through being immunosuppressed, have persistent infections. So large viral populations get to encounter sub-protective levels of antibody for longer periods of time. That, of course, accelerates viral evolution. There are some therapeutic modalities that, in principle, could affect viral evolution, allowing the virus to experience antibody therapy. And in particular, convalescent plasma therapy has been given to tens or hundreds of thousands of people with essentially uncharacterized plasmas with poor neutralizing activity. That obviously isn't a good idea if your goal is not to train the virus to evolve resistance to antibodies. But a criterion that is going to feature heavily in the talk is antibody diversity. What different kinds of antibodies does the virus encounters? If it turns out that everybody made exactly the same antibodies to the virus, then it would be essentially trivial for the virus to learn about those antibodies and escape those antibodies. But if everybody made different antibodies, then acquiring antibody resistance in one individual would in part no advantage to encountering antibodies in a different individual later in the transmission chain. And so, as we'll see, antibodies are quite diverse, but not absolutely unique in each individual. So we, in collaboration with Michel Nussensweig's lab in particular, we've been studying neutralizing antibodies for quite some time now. So this is sort of a sort of representative of what we've found from individuals that have been either infected or vaccinated. We're studying in particular antibodies that bind to this part of the spike protein, the RBD. We do that because those are really the most potently neutralizing antibodies, and not completely, but they do constitute a significantly large fraction of the neutralizing activity in the blood of convalescent or vaccinated individuals. And so what this slide shows in this circus plot, each slice here represents an individual who's either been infected, so convalescent individuals in green, or individuals who have been vaccinated with Moderna or Pfizer mRNA vaccines. And each slice has a black portion that represents memory B cells in that individual that have expanded. And so when the SARS-CoV-2 RBD reactive memory B cells in that individual are sorted, you can find essentially the same memory B cell multiple times in that individual because it's divided and expanded. Those are what we call expanded clones. The white portion of these slices are memory B cells that are found once. So they're singletons or unique. And the lines here connect memory B cells, those with essentially the same antibodies in different individuals. So they've undergone the same types of recombination events to generate unique antibodies, but we find those antibodies turn out not to be unique. They are found multiple times in multiple individuals. And so what this tells us is that there is at least a degree of homogeneity in the antibody response between individuals. So if you imagine that transmission chain again, it's quite often that the virus is going to encounter the same antibodies in different individuals over and over again. And that's worth bearing in mind when we think about how the virus might evolve. Now, these antibodies have various different potencies. You're going to see quite a number of charts in my talk where we plot the IC50, the potency of the antibody on the y-axis, and the lower down it is, the more potent it is. And in various individuals here, you can find that there are each individual antibody cloned from that individual has a characteristic potency that can be extremely variable, from extraordinarily potent down here, less than 10 nanograms per mil IC50, to essentially non-neutralizing. And it turns out that basically everybody that has been studied can make these potent antibodies, but the plasma neutralizing titer, how potent blood plasma from that individual neutralizes the virus, really isn't related to the potency of the individual antibodies. It's really related to how much of those individual antibodies each person is making. So this person, for example, with a rather unimpressive plasma neutralizing titer still has potent antibodies in their B cells. They're just not manufacturing large amounts of them. And they're really no different in that respect, no different in the types of antibodies that are made to this person, for example, very potently neutralizing, but the range of individual antibodies is similar. So like others, we've cloned some of these antibodies, and at Rockefeller, a couple of these antibodies are now in clinical development. They're in phase 2-3 trials for use as a subcutaneous injection. They are so potent that really rather small amounts of these antibodies can be given to individuals to elicit what we expect to be therapeutic and protective levels in their circulation. And so partly for that reason, but also to study the effects of vaccination and convalescence, we've been interested in resistance to antibodies for quite some time. So I introduced this last time, but let me remind you of a particular tool that we use that's going to feature prominently throughout the talk. And it's using an engineered virus that we use to select for resistance mutations, really to try and understand how resistance might manifest itself in the context of therapy and evolve in the context of convalescence, convalescent infection and vaccine breakthrough. So what we're using is a vesicular stomatitis virus where its spike protein has been replaced by that of SARS-CoV-2. This virus also contains a reporter gene, so we can nicely follow infection, as you'll see in a second. Unlike SARS-CoV-2 itself, VSV doesn't have a proofreading polymerase. So when we grow this virus, we can generate large, diverse populations, 1 million variants or more in a rather small volume of cell culture fluid. And we're doing this in the context of 2 different isolates. So you're going to see these terms 1D7 and 2E1. They're just 2 different stocks of the same VSV, diversified VSV population. And what we do is we generate diversity, mix those diverse populations with an antibody. First part of the talk is going to focus exclusively on monoclonal antibodies. And then we put that mixture on a cell culture, the non-neutralized fraction, the variants that have escaped neutralization by the antibody will selectively replicate, and then we will harvest those viruses from the supernatant. Sometimes we sequence them, sometimes we isolate the individual viruses to characterize their neutralizing properties. We're going to be doing both as we go through. This is just to give you a visual idea of how the experiment looks. This is a 12-well plate where we've put 1 million infectious units of that viral population onto cells. In the absence of antibody, you get about 1 million infection events, each one represented by a little green speckle here. If you have a neutralizing antibody, then most of that's blocked, but you'll see there's the odd little group of green cells here. If we just zoom in on this part of the plate, you'll see what's a nascent plaque. So this is an individual virus that's managed to infect the monolayer and is beginning to spread because it has a mutation that confers resistance to this particular antibody. And so, as I say, we've been doing this for quite some time now, but one of the first mutations that we found was this one, so-called E484K, which for a while late last year was the sort of the scary mutation that the journalists like to cover. It's quite surreal, actually, to take phone calls from journalists asking me, tell me about this mutation E484K. And the obsession of the media with individual amino acid substitutions for a time was quite interesting. Nevertheless, it is an important mutation because it does confer resistance to a rather commonly elicited class of antibodies. One of those antibody types that's connected by those lines that you saw in the circus plot a few slides ago. So these are 3 antibodies, C121, C144, and C135. 2 of them confer. 2 of them, they lose their activity in the presence of E484K. So here you see neutralization of the parental virus, and here you can see neutralization of the E484K mutant. So this antibody retains full potency against that mutant. These 2 antibodies, again, they're both very commonly elicited antibodies, have completely lost potency against that variant. So, as I say, we started talking about this last summer. At that point, sort of dogma in the field was that coronaviruses did not really varies so much in terms of acquiring antibody resistance. And I won't name the rather well-known virologist whose reaction to our work at that time was, "I'm not worried about this at all." I think time and experience has told us that we perhaps should have been worried about antibody escape a little sooner than we were. Anyway, by now we've done many of these types of experiments trying to select resistance in the laboratory to neutralizing antibodies and attempted to relate that to what we see going on in nature. So here's one such experiment where we've taken a bunch of 18 antibodies that were cloned from people who have received vaccines. But as I told you, they're essentially the same types of antibodies that are found in convalescence. Done exactly the experiment that I've just described to you, and then sequenced the virus that survives just one passage in the presence of antibody and look for what mutations are present in that virus that's passaged through that antibody. And what's shown here are the percentage of viral sequences that have each of these mutations listed across the top. And so just by looking at these 18 antibodies, we've gotten quite a slice of the so-called the mutations that have appeared in variants of concern in recent months. So you'll note in particular mutations at K417, E484, and N501. Of these 18 antibodies, 15 of those select for mutations at those positions. And those, of course, are the substitutions that have occurred in the P1 and the B1351 strains that have come out of Brazil and South Africa, respectively. There are some other mutations that have come and gone in various variants that have cropped up throughout the world. So N439 and 440 represent variants that rose to prominence in Scotland and Western Europe before dying away, and also in India before coming up and then dying away. The L452R substitution that appeared in the so-called California variant and is now present in the Delta variant that's swept across the world. Other variants haven't yet hit the headlines, but have clearly appeared at some frequency in variants that have cropped up in nature. It's particularly interesting to go through sequences that have been acquired from wastewater samples, and you can find many of these substitutions in that context. So by now, whenever a new variant of concern appears, we are completely unsurprised by the list of variants that appear in the receptor binding domain of those variants of concern. In just about every case, we have encountered said variant in some antibody selection experiment or another. Okay, so those substitutions in the RBD obviously constitute part of the binding sites for antibodies. They also sometimes overlap with the receptor binding site, which has caused some interesting discussions, controversy, confusion about what exactly is driving the evolution of those substitutions. You will deduce from my comments that I'm of the opinion that antibodies are a large part of the selective pressure that is driving those substitutions. But it is true to say it is at least conceivable that part of the evolutionary pressure is modulating interactions with the ACE2 receptor, which basically binds to a similar place as many of these neutralizing antibodies. So Christopher Barnes and Pamela Bjorkman, with whom we have worked very closely, have solved structures for many of these antibodies in complex with the RBD, show clearly that the the mutations that are arising are in the antibody binding sites. They've come up with a classification for neutralizing antibodies that target the RBD based on sort of the angle of approach, what the particular epitopes are, and whether they bind the RBD in the up or down conformation that can be seen in the three-dimensional spike. And so most of these antibodies fall into class 1, 2, or 3, as I say, based on structure and epitopes. And the variants of concern variably affect the binding of one or more of these three really quite common types of antibodies that are elicited against the RBD. And so one can think about this in a somewhat straightforward way, that virus is passaging from human to human. There are commonly elicited antibodies in a subset of those humans. That subset is obviously increasing as more people get infected and more people get vaccinated. And increasingly, that population constitutes an environment that puts a selective pressure on the virus to acquire and maintain those substitutions. And so I think it's important to sort of recognize that the types of mutations that occur and become dominant, there's been some talk of so-called convergent evolution, and that's absolutely what's going on. But there's also been talk of how this might indicate that viral evolution is constrained, that these mutations keep coming up because they're really the only mutations that the virus can tolerate and still propagate. I absolutely don't believe that to be true. I think what's happening is that the same mutations are coming up repeatedly is because they're encountering antibodies that are particularly common, okay? Antibodies that are found in many different individuals. And so one, in principle, could think about stopping there. What's happening is the virus is encountering antibodies, it's evolving, and eventually escaping those antibodies. But the situation in life is clearly a bit more complicated than that. And one of the reasons it's more complicated is because it's not just the virus that evolves, antibody evolves too. Okay, so when a B cell encounters an antigen in the form of a virus or a vaccine, the B cells grow and divide before they start making antibodies or become part of the memory compartment. And during that process of expansion, the antibody genes are mutated. Okay, so most of what I've told you about now so far has been focused on antibodies that we worked on quite early in the pandemic. The first sets of people to be infected or take part in vaccine trials. And what we find is that those antibodies, or I say Michel Nussensweg group finds that those antibodies have rather low levels of mutation. They haven't evolved very far from their so-called germline configuration. That's in part because the germline of some antibodies is actually particularly good at recognizing the SARS-CoV-2 spike protein. But if we let things mature for a while and look at antibodies some months after people have been infected, in this case, we find that the antibodies have acquired a significantly higher degree of mutation. So that mutation really requires that antigen be persistent. It doesn't necessarily require that infection be persistent, but it does really require that those B cells are exposed to antigen in order to continue to grow and divide and mutate their antibody genes. I'm just showing this for interest. A collaborator of ours, Saurabh Mahandru at Mount Sinai, has found that in a subset of individuals, he can find virus in the epithelial cells of the gut in a subset of individuals up to 3 months after infection. So even if there isn't, so at least in the subset individuals, it does appear that infection is perhaps more persistent than we might imagine. But even if infection isn't persistent in many, it does appear that at least antigen is persistent, sufficient to drive evolution of antibodies. And so I'm going to spend sort of the rest of the talk talking about the consequences of the evolution of those antibodies. And we've looked at it to varying degrees of detail, depending on different antibodies. Obviously, people make many antibodies. You can't study the evolution of every antibody lineage, but I'm going to show you a few examples now of what antibodies can do as they evolve, as their memory B cells grow and divide and mutate their antibody genes, and how that can affect the properties of the antibodies, really the antiviral properties of antibodies. So we've focused in some detail on 6 antibody lineages from 5 individuals. And so what we're really doing is comparing individual antibodies that were cloned soon in convalescence, 1.3 months after convalescence, and then later 6 months after convalescence. And I'm going to show you, these tables are a little bit dense, for which I apologize, but they're not so difficult to digest. What we're basically going to be doing is comparing antibodies isolated early or late in convalescence. The antibody designations here. This is the antibody concentration that was used in an experiment. And this is one of those selection experiments where we're selecting our recombinant virus for a particular number of passages, and then we've got the 2 different populations, okay? And then on the top of the table, what you're going to see are the mutations that arise in those passaged viral populations, and that's shown as a decimal fraction of the total, and the redder it is, the more frequently that mutation arises. And so what I want you to compare then is what happens when we select virus in this early convalescent antibody, and then in essentially the same antibody that's undergone a few months of sequence evolution itself, okay? So in this particular case, the early antibodies, they are clonal relatives. They come from the same parental B cell. Antibodies from those cells select for this mutation. The more astute of you will recognize this mutation as one that's occurred recently in the Delta variant that's spanning the globe at the moment. That's the dominant mutation selected by this antibody, and you can see where it is on the RBD here. If we do the same experiment with an antibody that's found later, it selects different resistance mutations, okay? Very closely spatially present, close in space to T478 on the RBD, but nevertheless, the preferred route of escape from those antibodies changes from the early convalescent to the late convalescent. Similar sort of thing here, but slightly different outcome. So in this case, the early antibody, when we select, there are various options for escape from that antibody. E484 is one of them, and Q493 is another. And the virus sort of has a more or less equal probability of choosing one or other of these mutations to escape that antibody. But if you then use the later antibody, what you find then is that you never see the Q493 mutation. It's all E484K, as if the virus has made the decision that that's the preferred route of escape from these later antibodies as opposed to the earlier antibodies. And we can sort of see why that is when we do this experiment. This is a different experiment. What we're doing here is taking a panel of pseudotype viruses. These are viruses where we've pre-installed each of these substitutions on the x-axis, and then we're measuring the potency of the antibodies on the y-axis. And so again, the higher up the y-axis you are, the less potent you are. But for wild-type virus, all of these antibodies are very potent. IC50 is around 10 nanograms per mil. And that's true for most of the mutant RBDs that we use, except these. So C144, the early antibody, cannot neutralize viral pseudotypes with any of these substitutions, okay? So that actually quite nicely defines the epitope of the antibody, but also tells you there's various options that the virus can take in order to escape neutralization by this antibody. Conversely, if you use these later antibodies, most of these substitutions don't confer resistance. And in this case, only substitutions at E484 can confer resistance to these antibodies, mimicking the result we got with the VSV experiment. This is really a manifestation of affinity maturation. As the B cells stimulated by antigens grow and divide, they're selected for B cell receptors that bind tighter and tighter to the cognate antigen, and we can measure those affinities to the 1.3 month antibody in the solid line against the wild type RBD in blue. That's here. Okay, and then the later antibodies, you can see they've acquired higher affinity, both for the wild type RBD, but they've also acquired affinity for the this particular mutant, the Q493R, that doesn't confer resistance. They haven't acquired affinity for E484K. That again is repeated here. So essentially, this narrowing of the spectrum of mutations that can cause resistance is sort of a side effect of affinity maturation, the antibody binding tighter and tighter as it evolves to the cognate antigen. And so in collaboration with Christopher Barnes and Pamela Bjorkman, we've investigated some of these antibody maturation events in atomic detail using structural analysis. And so, as you can see, the C144 early antibody and the later antibody, they have basically the same configuration in how they interact with the receptor binding domains on the spike protein. But at the atomic level, there's differences in detail. So this Q493 mutation, for example, that causes complete resistance to C144. While it does make contact in the mature antibody, it's less pivotal for the ability of this antibody to bind to the cognate antigen. And so therefore substitutions at this position don't result in loss of binding and loss of neutralization for the mature antibody as opposed to the less mature antibody. Another pair of antibodies where similar but even more striking situation occurs. Again, we've got an early and a late antibody. The early antibody, any one of these substitutions can confer high-level resistance to the 1.3 month antibody. None of those substitutions can confer resistance to the later antibody that's its sibling, that's just a more highly mutated derivative of it. In fact, with this C549 antibody, the late antibody, we see a new type of phenomenon. So again, this is the results of a VSV selection experiment. Again, with the early antibody, with high levels of antibody, it's quite easy to get escape, and the virus can choose a variety of different substitutions in order to escape that antibody. We try to do the same experiment with the later antibody using the same concentration of antibody. The virus can't escape. Okay, we totally fail to generate a viral mutant that escapes that antibody. What we find we have to do is to substantially reduce the amount of antibody and do more passages. Keep passaging the virus in lower concentrations of antibody. And then what we tend to get are viruses not with single substitutions, but what we tend to get are viruses with multiple substitutions, most often 2 substitutions. And so for this particular antibody, it actually turns out that you need 2 substitutions. And there are various options of which pairs of substitutions to use. But you do indeed need more than one substitution to escape that antibody. That's shown here. So here's C548 and its derivative C549 with the infamous E484K substitution. Complete resistance here only a sort of a fairly minor degree of resistance here. But if you combine that E484K with another substitution, Y549, then you get more substantial, albeit not complete resistance to that antibody. And then again, another pair of substitutions, proximal but not identical, can confer substantial but not complete resistance. Similar story with another set of antibodies, this time recognizing a completely different epitope. In this case, C98, it's a really bad antibody. It's poorly potent against SARS-CoV-2 and all these mutations, but its mature derivative now becomes quite potent and apparently resilient to all of these substitutions in the RBD that we throw at it. But again, if we do the VSV selection experiments, again, we have to back off on the concentration of the antibodies to begin to select resistance. Our standard conditions don't allow for resistance to occur. But if we use smaller amounts of antibody and allow more passages, then we get double mutants that have at least some degree of resistance as compared to the single mutants that don't, both here and here. And those, at least some of those single mutants, confer complete resistance to the parental antibody. And so by sort of repeating this story over and over again, the impression that we're getting and what I'm conveying to you is that as these antibodies mature and mutate and evolve and bind tighter to SARS-CoV-2, they become more resilient to the mutations that would otherwise cause resistance. And that obviously has important consequences as we'll go into. Now, I've highlighted a number of individual examples here. We haven't obviously investigated all antibodies in atomic detail, but Michel and his group have cloned literally hundreds, I would even say now thousands of individual antibodies from SARS-CoV-2 convalescents. And we've measured the potency of neutralization of just about all of those antibodies. And so we can begin to arrive at conclusions about the behavior of antibody populations as a whole. So if we look at all antibodies from a cohort cloned at 1, 6, and 12 months, you can see as a population, those antibody clones are tending to get more potent with time. If we divide them up into different types of antibodies that we only ever encounter once, antibodies that are part of expanded clones at any time point, or particularly antibodies that are persistent where we find expanded clones both early in convalescence and much later in convalescence, what you can see is the potency of those antibodies is clearly increasing. And this set of 12-month antibodies, their average potency is really quite impressive. 35 nanograms per mil is really an impressive number for neutralization by a single antibody. And again, if we look at the spectrum of mutations that can confer resistance to those antibodies, we arrive at a very robust conclusion that these individual antibody lineages acquire breadth over time. What I mean by breadth is the ability to neutralize a mutant virus that its predecessor could not neutralize. Okay, so this is 15 pairs of monoclonal antibodies cloned at 1 month and 12 months after infection. And what's shown here are the various IC50s. White is good, red is not good. And of those 15 of them acquire the ability to neutralize a mutant virus that they couldn't neutralize at the beginning. Only 3 out of 15 of those antibodies could neutralize this virus we call Ken. It has this set of mutations. Those were the mutations that were found in the 351 and the P1 strains from South Africa and Brazil that we were concerned about for a while. whereas 10 out of 15 of the mature antibodies can potently neutralize these mutants. And if you just go through, obviously we don't have time to look at everything in detail. If you just look at the pairs of antibodies, you can see that in just about every case, the 12-month antibody has greater ability to neutralize multiple different mutant viruses and do so with greater potency. So what I hope I've hammered home by now is that the antibodies evolve and they evolve in a way that's advantageous to us with time, okay, after people have been infected by SARS-CoV-2. Now, what we've most recently found and published is that the behavior of those individual antibodies is reflected in the ability of plasma from individuals to neutralize virus. And that it's sort of revealed in, I would say, somewhat spectacular form if you take people who are convalescent and then boost their antibody responses by mRNA vaccines. And so these are plasma neutralizing titers for individuals investigated at 1, 6, and 12 months after infection. And then a subset of these individuals, based on the timing of the vaccine rollout, a subset were vaccinated between the 6-month and the 12-month time point. That's basically earlier this year. And so if you compare those individuals at 12 months, those that hadn't been vaccinated, and so this is the sort of the natural longevity of the antibodies from convalescent infection. If they're vaccinated, you get a huge increase in titer, 20-fold against the original strain. But perhaps more importantly, this decrement that one gets when one uses variants of concern, in particular the B351, substantial fraction of individuals don't neutralize this virus well at all, at least in convalescence. But if those convalescent individuals are vaccinated, then all of them neutralize this variant, and all of them neutralize this variant with really impressive potency. We're talking titers in the range of 10,000 to 100,000. Actually, the average in this case is 11,000. But for all of these variants, we get these very impressive neutralizing titers. And what we think this is really reflecting is the antibodies having matured and then being the memory B cells being given a kick with a vaccine to generate these very impressive neutralizing characteristics. So I've spent most of the time talking about individual antibodies and their properties, but of course antibodies don't act as individuals. They act as I've just shown you, as components of a mixture of antibodies. And we've been also very interested in understanding what those different antibodies exactly are, focusing not just on the RBD, but thinking more globally about the spike protein. And really asking this question: what and how many different antibody specificities are responsible for the activity of plasma? Okay, is it just the antibodies that we focused on, those class 1, 2, and 3 antibodies, or is there more to it than that? The answer is yes, but I'll show you how we arrive at that conclusion. And so the tool that we've used to try and get at that question again is this recombinant virus, where we can use the virus to try and tell us where antibodies are recognizing on the spike protein by selecting for resistance. But this time, instead of using monoclonal antibodies, which in many cases is really quite straightforward to select resistance, what we're doing this time is to select with polyclonal antibodies, plasma. So these are plasmas taken from our cohort here at Rockefeller early in convalescence, so 1.3 months on average after they're infected. And we took 27 individuals who had the highest titer of plasma antibodies at that time. The goal being to really understand what the antibodies are, the good antibodies that are constituting those high neutralizing titers in that subset. So essentially what we do is the same thing. We take a plasma dilution that's tailored for each individual, incubate it with a viral population, and then select viruses that survive. Because this is a very much more complex mixture, what we typically have had to do is passage multiple times in the presence of plasma, sometimes increasing the amount of plasma, sometimes decreasing, essentially to try and titrate the plasma so we're at the optimal level to select for mutations that are conferring resistance to the mixture of antibodies that are present in that plasma. And so this represents sort of an amalgam of the results that we've obtained. So what we've got here represented on the x-axis is the spike protein sequence, the spike codon. And then on the y-axis is how frequently we find variation at that position, okay? And we've pooled all 27 of these selection experiments into one graph. If you look at them individually, it's really hard to discern patterns, but when you put the data all together, you start to see what I'm about to show you. And that is that plasma tends to select at particular positions in the spike. Not every plasma selects at every position, and some, the virus completely fails to grow. But as a population, this group of 27 plasmas clearly can impart evolutionary pressure on particular parts of the spike protein. Now, if we compare that variant frequency that's driven by selection by neutralizing antibodies in plasma, and we compare that to naturally occurring variation, global variant frequency in the GISAID database that by now has, I think, more than 1 million SARS-CoV-2 sequences around the world. It's not perfectly concordant, but there's clearly a significant degree of overlap between what we select with plasma in the virus and what is rising to the surface to to become present at higher frequency in naturally occurring variation, okay? And these gray stripes highlight the parts of the spike protein where that's most evident. We can represent that variation on a three-dimensional representation of the spike protein, and where it's red, we see more variation selected by plasma in our in vitro cultures. And in red here is represented global variation in spike protein sequences over the course of the pandemic. And I hope you'll agree that while they're not exactly the same, there's a significant degree of concordance. To us, that strongly suggests that at least part of what's driving natural sequence variation in SARS-CoV-2 is selection by antibodies. Now, in addition, we can take these selected populations and clone individual viruses having been passaged in plasma. And we've cloned 38 of these different mutants that have been selected by plasma. Occasionally, they overlap with the viruses that we select using monoclonal antibodies, as you'd expect, because those antibodies and those plasmas come from the same individuals. But if we take these mutations that have been selected in plasma and ask, are they resistant to the plasma that was used to drive their selection? The answer is almost invariably kind of. They're a little bit resistant. You can see this one, right? There's a rather minor effect of this mutation on susceptibility to neutralization by this plasma. Sometimes we have bigger effects. E484K, for example, generally has a larger effect. But in general, the individual mutations that are selected by plasma have a rather modest effect on neutralization by that plasma. But if we start to do this experiment where we combine mutations that were selected by different plasmas, combine them into a single spike, and generate what we call synthetic polymutant spike proteins, we start to see bigger shifts in sensitivity to plasma. So here are 3 examples of one of these, what we call polymutant spike proteins that has 13 different mutations put into it. You can see there's somewhat modest changes in susceptibility to these two plasmas, rather bigger change in sensitivity to this plasma. And if we do this with all 27 plasmas, we can see that the individual mutations, there are in just about every case a reduction in the neutralizing titer, but in just about every case it's quite modest reduction in neutralizing titer. But if we pool a subset of those mutations, and they were chosen sort of empirically, then you find bigger changes in neutralization. So we made a couple of these sort of different polymutant spikes, each with 13 substitutions, based on somewhat of our best guesses of what we saw during those plasma selections, to try and generate spikes that had a degree of plasma resistance. So here's a couple of examples, and you can see in red the neutralizing titers against those what we call polymutants is clearly reduced as compared to the neutralization of the parental wild type spike. And the magnitude of that resistance is sort of in the range that we find with natural variants of concern or variant of interest spikes. So they're more resistant than, for example, the B117, less resistant than the B3351, which have, in some cases, fewer, in some cases, similar numbers of substitutions in those spike proteins. But reasoning, if we could generate a spike protein that was completely resistant to convalescent plasma and completely resistant to plasma from vaccine recipients, if we could do that, we would have identified essentially all of the neutralization targets on the SARS-CoV-2 spike protein. And so we went back through our plasma selection experiments. We looked again through the mutations that occur naturally that confer a large degree of neutralization resistance, particularly in this B351 strain. We went back through our monoclonal antibody selection experiments and picked out 20 mutations that we thought were most likely to generate some kind of antibody resistance, and generated this spike protein that we call PMS20. And in fact, this spike protein with 20 substitutions is nearly completely resistant to most of the plasmas from randomly selected convalescent donors and vaccine recipient donors. Okay, that tells us that these 20 substitutions define most of the targets of neutralizing antibodies in randomly selected convalescents and vaccine recipients. And so that is, I think, a quite important piece of information. It sort of tells us what the genetic hurdle is that the SARS-CoV-2 has to clear in order to get resistant to these plasmas that are elicited by convalescents and by vaccines, okay? Because that's the type of immunity that the virus is encountering for the most part currently. Now, interestingly enough, if we take those special plasmas, those plasmas from convalescent individuals who have been boosted by vaccination, so those antibodies have been allowed to mature and then boosted by vaccination, then even this highly resistant spike protein is quite well neutralized by plasmas from those individuals, with titers typically of 1,000 or more. So that really underscores how impressively different these matured boosted antibody responses are. And we decided to really see how far we could go with this. So we looked at some sarbecoviruses that are not SARS-CoV-2. These are in principle pandemic threat coronaviruses that we haven't yet seen. This is basically a representation of the spike on the x-axis and how similar it is to SARS-CoV-2 on the y-axis, and you'll recognize, for example, RATG13 that's very closely related to SARS-CoV-2, except in the RBD, a pangolin coronavirus from Guangdong, which has a very similar RBD to SARS-CoV-2, different in other places, and then some more, very much more divergent viruses, the original SARS, and then this bat coronavirus that have, up to 50% amino acid divergence in the NTD, 40% in the key parts of the RBD, and greater degrees of diversity here. And what we find, in fact, is that these plasmas from these vaccinated convalescents are actually pretty good at neutralizing these really quite diverse sets of viruses, some better than others. So this one, whose RBD is very much like SARS-CoV-2, very, very potent neutralization. But even SARS-CoV and WIV16 that are, as I showed you, 40 to 50% divergent in the relevant parts of the spike protein, we're still getting titers in the 10 to the 3, 10 to the 4 range. That's summarized here. So here we've got neutralizing titers against the original strain for a group of convalescents who were then later vaccinated. And you can see they have extraordinarily high titers against all the circulating variants of concern, including Delta, Beta, one you haven't heard about yet from Angola. And they also neutralize these diverse sarbecoviruses. And in fact, titers against SARS-1, the worst of these, SARS-1, the titers there are about the same as convalescent titers against the SARS-CoV-2. So to my mind, this type of result really represents the future of what we should be aiming for in vaccination. Can we achieve this kind of breadth using vaccine regimens that would enable us to deal not just with currently circulating variants, but likely with most future possibilities in terms of variants, and even perhaps provide some protection against the pandemic threat coronaviruses that we have yet to encounter as a human population. So let me quickly summarize. So SARS-CoV-2 spike protein elicits potent neutralizing antibodies in convalescents and vaccine recipients that have a degree of homogeneity, similarity to each other. Those antibodies have exerted evolutionary pressure on SARS-CoV-2. The natural variation that we see in global populations seems to be concentrated in the same place that we select for resistance using antibodies in the laboratory. About 20 spike mutations can confer nearly complete escape from most of those antibodies, at least the ones that are elicited early in infection or vaccination. But maturation of those individual antibodies can increase affinity, potency, and perhaps most importantly, constrains the options for viral escape and impose a higher genetic barrier, the requirement for multiple mutations for escape to occur. And if we vaccine boost this matured antibody response, it really is impressively broad, capable of neutralizing SARS-CoV-2 variants, including synthetic variants, where we've shown that 20 mutations can confer nearly a complete escape from those early antibodies. That is defeated if we allow the antibodies to evolve and then boost them. And then, of course, the key question is, can we mimic this impressively broad antibody response with the vaccination tools that we have currently and in the future? All right, sorry, I've gone a little over time. Let me quickly thank the people who I've worked with on this, in particular my wife and colleague Theodora Hadzianu, who's an equal partner in all of this work. It's really, in my lab, been spearheaded by 3 postdocs, Frauke Mutsch, Fabian Schmidt, and Jiska Weisblum, who since the beginning of the pandemic have really been working like heroes to bring these stories to fruition. Another additional group of people in my lab, Magda, Fengwen, Daniel, Justin, and Eva, also working hard on these antibody questions. Everything I've talked to you about has been part of a very close collaboration with Michelle Nussensweg's lab, who do all the antibody cloning, and also have a clinical group led by Marina Kaski and Christian Gabler, who recruited, see all the patients, get the plasmas, the B cells, and the material for the antibody cloning that Michelle's group do. Charlie Rice and his group have been involved in some of this work. And I mentioned on a couple of occasions, we're frequent collaborators with Pamela and Christopher, particularly with the structural parts of the work. Thanks you very much for listening.
Thank you, Paul. Thanks for the great talk. And of course, thanks for ending on that optimistic note that vaccine boost elicits this broad response. It's kind of what we wanted to hear at this point. Well, a great talk always brings in a lot of questions. So without further ado, let's dive right into the audience questions. Audience, a reminder, please feel free to send in your questions via your Q&A box. And in the meantime, Virginia, you have questions, so please unmute yourself and ask away.
Hi. Maybe as a basic question, I was wondering how much does the diversity of antibodies that are elicited by vaccines is reduced compared to sort of a natural infection? And is there any difference between the types of vaccinations that are currently out there? And then a second question is, I was really surprised by the fact that across the population, you only see certain variants of antibodies. And do we know if there's a genetic basis for this? So do families tend to produce sort of more similar antibodies, and what drives this? Thanks.
Right. So in terms of the vaccine response, what we can say is that early after vaccination and early after infection, there's really quite a degree of similarity between the antibodies that are elicited. Okay. I should say we've spent essentially all of our time thus far we're now beginning to diversify our looks on people who have been vaccinated with the mRNA vaccines. Okay, and the people who were infected with the early strains of SARS-CoV-2. We're just beginning to get people who have been infected with the more recent variants to look at their antibodies. Of course, they're going to be different, right? They're going to they've been exposed to a different antigen. And what the difference will be for different vaccines, we don't yet know. Now, how the antibodies are going to evolve over time in response to vaccination, will we get this same maturation, diversification, yet to be seen. We're working on that now. There'll be a paper submitted quite soon. Now, as to individual variation in what types of antibodies are made, clearly some of the antibodies you can find in multiple different individuals, but of course there are a whole set of antibodies that are unique to different individuals. To what extent that reflects genetic variation among people, and to what extent that it reflects chance, stochastic events, we just don't know at present. There's just, if you think what the repertoire is inside a single person, it's amazing to me that you ever find the same antibody in multiple different individuals, but you do. Okay, how human genetics affects that, I'm afraid I just can't comment at this point because I don't know.
Thanks.
All right. Thanks, Paul. Thanks, Virginia. Our next question is from Alfredo. Alfredo, go ahead. Ask your question.
That was a great talk, and I don't tell that very often to people that go over time. It could have been even longer. So the questions that they
Let me apologize again.
No, absolutely. It was fantastic. I love it. So the two questions I have is, one is about persistence. You mentioned that there might be persistence of the virus, but I thought maybe that is in contradiction with the decline in antibodies that is often seen in infection and vaccination. So that would be my first question.
Yeah.
Yeah.
So no, you're right. It's clear that the overall amount of antibody is declines after infection and vaccination. Sort of at the same time, at least in the infection context, as it's diversifying. Okay, so the overall, you have less antibody, higher affinity antibody. So if I just measured all the SARS-CoV-2 specific immunoglobulin, I'm sure that would be a more rapid decline than the neutralizing titer. But nevertheless, the titer does go down. And, some people think only memory B cells are important, but I happen to believe that actually circulating antibody also might be important in protection. So, having very low titers, very small amounts of potent antibodies is probably not good enough if those titers aren't enough to neutralize the incoming virus. Once you've mounted a sort of a recall response, then those antibodies could be strongly protective against the later course of disease. But to be frank, I'd be very much more comfortable having those antibodies boosted ahead of time.
Thank you. So the other question I have is that some colleagues, argue that probably this virus will keep finding escape mutants and more. And while I heard other people arguing that escape mutation will be limited by decreased viral fitness, so that the number of escape mutants will be a finite number. Based on the data that you presented, that you saw in the literature, what are your thoughts? Will this dog run out of tricks, or will keep finding new tricks?
Yeah, I don't think that the virus is intrinsically constrained by sequence space in terms of having to make a choice between losing antibody binding while maintaining ACE2 binding, for example. I mean, just look at SARS-CoV, right? Look at the diversity of sarbecoviruses, all using the same receptor, all infecting cells by basically the same mechanism. But you got 50% different amino acids in significant parts of the protein. There are parts of the protein, the fusion machinery, that are likely to be more functionally constrained. But there's really quite few antibodies that recognize that part of the protein that are strongly neutralizing. And so you have to ask, is that sequence conservation because it's functionally constrained, or it just hasn't been put under pressure? It's probably a little bit of both. My suspicion is this virus is going to continue to evolve, right? And basically the outcome of infection, or should I say the outcome of challenge, is really going to depend on how robust immunity is in each individual case. So, I think the virus is here to stay, and what probably the best, I don't want to overstate my opinions, because it's easy to convince yourself of something just by looking at what the situation with the seasonal coronavirus is, for example. That's absolutely a plausible outcome here, where we all have strong enough immunity to essentially contain the virus to our upper respiratory tract. Yet the virus is constantly evolving, finding individuals that have waned immunity, waned sufficient to propagate virus, but we're at a state where nearly all of us have sufficient immunity to ward off severe disease. I think that's the most likely outcome. But the virus isn't going to stop evolving because it's constrained, I don't think.
Thank you.
Thanks, Paul. So adding to that discussion, so since the virus is not constrained in the sequence space, so it's probably going to travel through the Greek alphabet, what is this? Oh, hopefully not. So, but then what is the state of, what's your take on vaccines? I mean, are the 2 doses that we have received, is that enough, or should we seriously consider booster doses? And if so, should the booster doses also be designed keeping the new variants in mind?
Okay, so I have to say everything I'm saying here is opinion, right? It's based, what I've just told you about, but my opinion is yes, we can and should get booster doses some months after the first 2 shots. But it raises a different problem. We can't even get half the population to take a single shot at the moment. So those boosters, those are for the half of us that actually believe that the vaccines are to our benefit. What that means in the long term, where we have half the population not vaccinated, could it be that the virus eventually evolves to defeat even the spectacular immunity that is potentially available to us? I don't know. But I think for the foreseeable future, my prediction is yes, based on what we're seeing with waning immunity to the initial 2 shots, a booster shot is going to be helpful, particularly against the variants. What we can say without a shadow of a doubt is that those 2-shot mRNA vaccines, while very effective against what we've seen so far, have in no way exhausted the capacity of the human immune system to tackle this virus.
Thank you. Okay, how are we doing with time? It's all brief 15 past and we still have audience questions. Do we have more time, Paul?
I'm fine.
Great. All right. So
since I went over time, it would be rather churlish of me to call hold.
All right. Lauren, do you want to go ahead and ask your question?
Sure, Paul. Amazing talk. I've got a couple follow-ups first to things that came up. So yeah, there's the question, what's the sequence space available to the virus? Is it finite or infinite? Is that really a good question? I have an even simpler question is, so individuals can definitely acquire breadth over time. How well do you think we will do at keeping that breadth? And do we, will we have a fall off such that the virus could, for instance, get to omega and then try alpha on us again? And it'll take us, a little bit to catch back up to alpha, and that some people would have fallen off and be vulnerable again.
Yeah. I think so I don't know any situation where evolution has been circular, right? So once alpha's been eradicated, and I think at some point it will be, we won't be seeing it again, but you're right, I think, in saying that evolution isn't constrained by the limits of the Greek alphabet. So we probably have to bring in another alphabet. It won't be alpha, but it might be. And to be honest, I'm trying to look into a crystal ball, and I'm not 100% confident about what I think about the future. If we do imagine a situation where and I think this is the most likely situation, we get to a situation like the human coronaviruses. I think, Jesse Bloom's done some very interesting and I think nice work recently, going back over decades, looking at what the seasonal coronavirus evolution is like. And it is directional, right? So it marches from one variant to another. And it hasn't, as for example, we see with HIV, gone with many different branches in all directions. It's as if the new strains replace the old strains, right? And a strain that's 30 years removed from an ancestral strain has really replaced it, and you don't go back to the original strain. So, at this point, because we have a big part of the world's population that has been infected, and yet more that haven't, we sort of still have to wait a little bit to see what the eventual evolutionary trajectory is going to be here. But my suspicion is it's going to most closely mimic what the seasonal coronaviruses do.
Yeah, I expect you're right. I mean, just with this virus infecting, having just a much larger N than any previous infection, I just wonder, if that's enough to support pools in different places, reservoirs to draw from. But
right,
I think you're probably right.
We're just going to have to wait and see, I think. Obviously, it's a situation where the virus goes off in multiple different directions. That is sort of a nightmare scenario. But even with what we know so far, that doesn't seem to be happening. I mean, Delta has done a pretty good job of replacing most of what was there before. It hasn't done it completely yet. We're only really still months into each new variant. So, this is going to play out over years, not weeks and months.
Yeah. So I had a similar question, but now at the microscopic level. So that's the macroscopic level of among individuals. In an individual, I'm curious about how the memory B cells evolve. So, I mean, you showed us that, the first wave would have, I think it was at 474, and then a subsequent wave would have mutations at 478. Do you think that the memory B cells are reverting 474 before committing to 478, or do you think it's the precursor population that was still resident that is being called on to make the newer mutations?
Right. So if I can rephrase your question, and please. I hope I'm doing this accurately, is it the virus evolving within an individual that's driving the diversification of the B cells, or is it the original strain and subsequent sequence diversification in the antigen doesn't matter? Is that what you're saying, or?
I guess I was just more curious about the level of the different cells, less on the cause.
So even though Michelle's lab sequences hundreds of antibody genes from each individual, I am not sure that you can really tease out those dynamics in great detail. Clearly, some clones expand, and some clones contract. We assume the contraction is because they're being outcompeted by higher affinity B cells. But it's true that sometimes new clones come up later. I honestly myself don't have a great understanding of the evolutionary forces that are other than antigen affinity that are driving B cell competition and evolution. Although they're clearly there, but you probably have to ask someone with greater immunological insight or perhaps more accurately, greater willingness to speculate than I on exactly what's driving the B cell evolution. We know that antigen affinity is crucial, but we also know that the memory B cells also include some less high affinity B cells, and Michel's group have a theory that that's sort of an evolved characteristic, that you have some not remarkably high affinity memory B cells, some diversity in that memory B cell population, precisely to deal with what you're suggesting, that variant viruses might better interact with the diversified memory B cells that aren't necessarily only optimized for affinity. There you go. I did speculate.
Perfect. Thank you. Should we? I think so. I think we have. Let's do another 3 minutes.
Sure. Okay. Okay.
Go ahead, and then let's call on Henri.
All right. So I have a naive question. You probably would have heard of this, maybe. So when you say that selection by antibodies, they drive evolution. So are we acting as incubators, especially the one among us who are like single vaccinated, or even people who are fully vaccinated, like as breakthrough infections? Are we in some way spurring or encouraging this virus to evolve?
That is a very interesting and a more difficult question than you might think. So really, somebody who has immunity, right? The impact on the evolution of the virus really depends on the frequency with which they act as a chain terminator in transmission, right? In which case they strongly constrain evolution of the virus, right? Versus whether they act as a propagator, just giving the virus a nudge in terms of evolution as it passes through. Now, the degree to which partially immunized individuals, people who've only had one shot, or even two shots and then get infected with a variant that's a little resistant, the degree to which those people act as propagators and just shapers of viral evolution, I think is unclear. I've been extraordinarily impressed by these sort of anecdotal cases of people who have immunosuppression that are persistent incubators of virus. And I think, some of these people have sort of issues having been transplanted or have congenital immunodeficiencies. But there are also quite a large number of people who have HIV infection who are quite immunosuppressed. And there's been the odd reports in each of those scenarios where you can see sequence evolution in those individuals that's impressive, particularly impressive to me, who's someone who's trying to do viral evolution in cell culture. When I see what happens in these individuals who have, obviously they have larger viral population sizes, they're huge incubators of virus. The amount of evolution that can occur in a single individual in the right circumstances is really quite remarkable. And it's possible, I think it's at least possible that those rare individuals are at least partly responsible for the variants of concern that we've seen. The degree to which partly vaccinated populations, such as existed for months in the UK, the degree to which they are acting as drivers of evolution, I'm less convinced about, but it's still a possibility and not something we should be pursuing as a matter of policy, as the UK did.
Fair enough. Thank you.
Yeah, Paul, thanks so much for your beautiful work and all your time. And I wish I wouldn't say see you in 8 months, but I think I have to.
All right. Thanks very much. I enjoyed it. Thanks for all the questions.
Take care. Everyone tune in
August
3 weeks, August 17th. There we go. Nate Grubau, Yale, and get the latest and greatest from there. Okay, everyone stay safe, and we'll see you soon. Bye-bye.